Hello, hello, hello.
Audible. Namaskar. Our speaker sir has joined us, so we are going to start our session. Namaskar and a very good afternoon to one and all present here. It gives me prodigious pleasure to extend my warm welcome to the day 4 of our EFDP session. I heartily welcome our esteemed guests, eminent speakers, respected faculty members, active participants to this EFDP program, entitled as Immersive Technologies and Artificial Intelligence in Pharmacy Education, Building the Future of Learning. On behalf of organizing committee, I Ms. Murtuza Singh, Assistant Professor of Bharat Pharmaceutical Technology, welcome you all once again. Today, we are extremely fortunate to have with us an astonishing and astounding speaker, Dr. Dharmendra Ahuja, who is Principal, Mentor, X-Form Software, Bureau for Health and Education Status, Upliftment, India. So now I request Mr. Arnab Majumdar, Assistant Professor of Bharat Pharmaceutical Technology, to kindly grace our speaker.
Thank you, Motoshi Madam. Good afternoon, everyone. It's my privilege to introduce today's distinguished speaker for our e-faculty development program, Dr. Dharmendra Ahuja, a highly respected academician and mentor in the field of pharmacology. Dr. Ahuja holds a PhD in pharmacology and brings with him more than 25 years of rich experience in pharmaceutical domain. He has served in diverse and impactful roles, from being the principal mentor at X-Farm Software, Bureau for Health and Education Status Upliftment India, to a guest professor of pharmacology at Rajasthan University of Health Sciences, Jaipur. He is also actively contributing as a member of Indian Pharmaceutical Association, a board member of JB Women's University, Jaipur, as well as an M-panel assessor with NAC and inspector with Pharmacy Council of India under Government of India. Dr. Ahuja's professional journey is marked by excellence, having previously been a fellow with both the Department of Planning, Government of Rajasthan, and the MHRD, Government of India. His outstanding contributions have been recognized at highest levels, receiving appreciations from Health Minister and Women and Child Development Minister of Rajasthan, as well as the Mayor of Jaipur, among many others. With multiple patents, numerous publications, and lifelong dedication to pharmaceutical education and research, Dr. Ahuja continues to inspire scholars, educators, and professionals across the country. It is indeed an honor to welcome Dr. Ahuja to this session, and I'm sure his insights will enrich us all. Sir, I would kindly request you to start with today's session. Thank you.
Now I request Dr. Ahuja sir to start the session.
So, first of all, I would like to thank the management of Bharat Technology for inviting me, respected principal, Dr. Shantanu Malik, all other learned academicians on the dais. And it's a really matter of pleasure that Bharat Technology is organizing this type of faculty development program for development of faculties in their skills. And it is very much needed because nowadays new technologies, AI and various new fields are coming. So it is very necessary to amalgamate these latest technology in the education. Because if we will amalgamate these technology in the education, we will deliver more to our students. And in the FDP, in the faculty development program, it is the appropriate point of time to include AI and such similar type of things for improvement of the faculty. Now coming to my topic today, I'll be sharing basically application of AI in preclinical pharmacology. So first of all, I'll be sharing this small PPT in which we will see what is AI, how it is applied in the preclinical pharmacology, and then if time will allow, we will try to practice some AI tools using X-Pharm experimental pharmacology software. So First, I'll share the PPT, then we will try to practice the software as well. I'm just sharing the screen. Is this visible?
Yes, sir, it's visible.
Yeah. So we will be discussing like application of AI in preclinical pharmacology. In the discussion, first we will be see like what is AI and everything. Then later on we will see what is virtual experimentation in pharmacology, what is virtual experimentation techniques, what are the softwares used in pharmacology, physiology, what are the advantages of virtual pharmacology. So AI, because everyone is using the word AI, we should actually know what is AI. Means AI is nothing but application of the software, but it is a, we can say, next layer of the software which have the ability to translate the spoken or written language, analyze the data, and based on that data, the AI have the advantage to give its recommendation. See, all of you must have used Google also and ChatGPT also. So in Google, whenever you search something, it gives the available web sources. But in ChatGPT, when you write a question, suppose in Google you will write what are the best you can say best any question, what are the best colleges in Jaipur, if you are writing. So Google will give you the list of best colleges in Jaipur. And in ChatGPT, you can write, I want to take admission in a NIRF rank or NAC ranked, this much ranked college. So please give me that college name. Now there, this part of this ChatGPT starts, it will search from that data and give you one particular result of that data. So AI is little bit advanced. AI has the power to analyze the data also. Means additional data analysis part is also added, then it is called as AI. So in preclinical pharmacology also, we will be using AI now. So we will see how in preclinical pharmacology this has evolved and this has came. So this prevention, first we should know how the government acts came and all. So Prevention of Cruelty Act was passed in 1960. And in that act, they told that harm to the animals should be avoided and film, charts, books, etc. should be used. And but see, in today's scenario, we are in the latest version, latest level. So here what happens, here the things have changed from film chart because we are in 2025. The act was passed in 1960. So at that moment, they have written model film chart. But if you will correlate it with today's scenario, it is software, it is the visualized software, it is VR, it is virtual reality also is coming a lot. It is AI in the teaching. So in this section, we will be seeing like what is AI application and all. So this was the background, and virtual experimentation in pharmacology. It's now not a new thing because since 20 years it has evolved like anything, and it is called as in situ pharmacology also. Sorry, in silico pharmacology also, because in silico means whenever you are performing a practical on the system, it is being performed on silicon chips, and those silicon chips is correlated with silico. So whenever you are performing any practical with a major role on computers, so that experimentation technique is called in silico technique. So this preclinical pharmacology using virtual software is also called as in silico pharmacology experimentation. See, number of now this pharmacology itself has your teaching part, application part. But as we all are means from medical background, we are here to understand pharmacological concepts. So I will not go in the technical part. I will go in the pharmacological application part, because developing software requires HTML, then Python and all other knowledges. That is another branch. But here we will see its application. So how we can apply the software in analyzing data, in understanding our concept of preclinical pharmacology. So XPharm software, it is a experimental pharmacology software and being used extensively by most of the colleges. And it is a Bureau for Health and Education venture. And so this is XPharm software history. Earlier in previous history, various academicians tried to prepare different modules, but Bureau for Health and Education has prepared this with the input of more than 100 of pharmacologists, and it's a long journey. And now it is established software which is being used by pharmacologists. So these are the number of experiments. The purpose of showing this number is like to say you how vastly it has grown. If you will see the modules, it is containing 87 group modules. 87 group module, it means it is covering complete your B.Pharm syllabus. So it is providing even MBBS syllabus it is covering, and it is providing veterinary coverage also, and it is also having physiology modules also certain to make the pharmacology module more understandable. Certain physiology module, in fact, Pharmacy Council of India has also included certain in their guidelines to understand some physiology modules. So it covers physiology as well as pharmacology. So the purpose of adding the modules was like how vast it is. These are few other additional things which are available. Exam mode is there. Then videos are there of each module, how to use. Then study material is there, how to use. So complete animal cruelty prevention package is there, this one. And Pharmacy Council of India has already told in 2014 that wherever possible, we should avoid using animals. Physiology software is also there that covers physiology modules. So basically, the Bureau is providing a broad range of modules which covers all the needs of pharmacy courses as well as medical courses as well as veterinary courses. And for whom, as I told, it is for pharmacy, veterinary, nursing, physiotherapy, whosoever is studying pharmacology, preclinical practical pharmacology, they should go through the software. How to get this software? Simply in Google, they can write down XPharm, experimental pharmacology software, or Bureau for Health and Education. If they will write, they will get the link for the software and they can go for it. The advantage is like it provides benefit to avoid use of animal. Many experiments can be demonstrated in a very short time, and it decreases the cost of maintaining the animal. And also the test reports are also available for the teachers at single click. So there are various advantages are also available with the software uses. So this was the brief preview, like what is this preclinical pharmacology, what is AI, what is prevention of cruelty to animal act, then why this had evolved, what are the experiments, what are the advantages, how to get this one, where it is applicable. So all these things we have summarized in a PPT. Now we will try to see some one or two module also. So I'll just log in into the software. Just hold 1 minute. I'll just open with a sample ID. We will to log in into the software, just we have to open the Google and there we have to just write down XPharm login. When we will click on that login page, we will get this login page. You can see it is having various affiliations as well, like AICT listing is there, Ministry of Education listing is there, Indian Pharmacological Society has also listed the software in their website. If you will click on in Google, you will write down, you will get this page, and when you will click on these links, you will get their website also have included the link, same link in their web pages as well.
Sorry to interrupt, sir.
Yeah.
Sir, the screen is not visible.
Screen is not visible. Okay, I'll once
Yes.
again share.
Okay.
Is it visible now?
Yes, sir, now it's visible.
Yeah, so this is the login page of the software. What I was discussing is like means there are other links in addition to PCI, other links are also there, like AICT link is there, then Ministry of Education link is there, Indian Pharmacological Society. And so when you will click on this AICT link, you will get the AICT web page where they have listed this particular software. Similarly, when you will click on Ministry of Education link, you will get the Ministry of Education website link where they have listed this software. When you will click on this IPS, Indian Pharmacological Society link, you will get the link where the same link of this software is available. So this shows the expansion, the deep reach of the software to various association it with the renowned agencies. Now we will try to log in into the software. So this is the login page. Now one sample ID, I will just enter it here. So I have entered one sample ID and we have logged in into the software. See now all these our syllabus experiments are there. Basically, it is covering pharmacology, additionally physiology module, and covers the need of all pharmacy as well as medical and means veterinary students as well. To see some experiment, we will click on this first one, Rota-Road experiment, which is very commonly used in pharmacology setup. So I have clicked on this Rota-Road experiment. See, this is the Rota-Road experiment setup. Now see, in this one, different sections are there, like it covers various sections like introduction, experiment. So when we will click on the experiment part, we will see the instruction. So complete instructions are there, like first animals are divided into two groups, then administer one group with the drug to be tested, other with the vehicle, then click on the rodent one by one, then put the rodents on the Rota-Road apparatus, then check, start the Rota-Road apparatus rotating at a speed of 25 rotation. Then when the animal fall, respective time we have to calculate. So all the instructions are there. Previously introduction was there, concept was there. Now suppose we are performing the practical, we will just click on administer. So this is the administer video is coming. Once the drug will be administered, automatically the animals will be divided into two groups. And we have to click on suppose I am clicking on vehicle treated group. So I have clicked on it. So these animals will be kept on the rotating rod apparatus, which is Rota-Road apparatus. Now we will see the falling time of all these three animals, and we have to note down that falling time in our, you can say this observation table. Just 1 minute. See, we have seen the falling time of all the animals. Now we will just write down that falling time in our observation table button. 55, 50, and 52. Now, similarly, we will do the same exercise for the drug treated ones. I have clicked now drug treated. I will enter for them also. Now, once again, I am evaluating the falling time for drug treated animal. We have to see the time in which the animal is falling. See, the middle one has fallen at 39. The rear one has fallen at 45. The other one has fallen at 42. So whatsoever their falling time is there, we have written it here. Now, that time whatsoever I am entering will be automatically visible in the observation table section. See, this is the observation table. What I have entered, it is visible. See, 42, 39, 45 was the time I entered. So this was the falling time of all the three animals. So we have seen the falling time for drug treated animals, diazepam treated, and we have seen the falling time for vehicle treated. Now, I have not mentioned free riding. We can ride free riding also. The number of time they rotates completely on the rod. So for that, I am just explaining. So I have entered one data in that. So now whatsoever data we have entered, suppose student is performing. Suppose I am a student and I have performed this one. So now I have to submit this to my teacher, my report. So I will enter my name here. Suppose I am entering my name here, Dr. Dharmendra Ahuja. Every student can write down their name after performing the practical, and then they will click on this start download. So this complete report will be downloaded. This report will be visible to you at the download section. See, this is the report. My name is there, and this complete whatsoever my readings were there, those are there. And after performing complete, I can go for conclusion part, like observed reduction in fall off time of free reading shows that diazepam 1 mg per kg produces reduction in motor coordination and decreases the muscle strength. So what the observation table which I just downloaded, the student can take printout of that and they can submit. Now this was in the tutorial mode. When they will do in exam mode, drug name will not be visible. Here diazepam name is visible. The same experiment is available in exam mode also. I'll just show you. Suppose I am clicking, I am going back. I'll click on exam mode button. In the exam mode button, I'll once again search that same practical, Rotarod practical. See, this is in the exam mode. Now in the exam mode, see here sample drug is written. Here drug name is not written. So when they will perform on the basis of their IQ, they have to give that which drug could be. The falling time can reduce also, can increase also. Then once again, they can take printout of their reports. So on the day of exam, they have to perform in exam mode. And on the day of normal class, they have to perform in tutorial mode. And they have to take printout of their reports and they can submit with their answer sheets or in their registers. So this one experiment I have shown to you. Similarly, number of experiments are there, and it is covering a great range of modules also. Additionally, we have created one more student teacher reporting panel is also there, where the student can create their IDs, teacher can create their ID. Although PCI is not asking for that because the PCI is asking to submit answer sheets or practical records. So in that case, the initial exam mode is useful, but for another saving purpose and all, we have added one more panel there. So additionally, supportive resources like supportive visual resources are there. Means how to how to handle the animals, what are the physiological salt solution. And additionally, when you will log out this one, you will get one and the login page. See, everything like how to. I'll just show you one minute. See, this is the where we started the practical. So there explanatory visual link is there and lab manual link is also there. So when you will click on lab manual, complete lab manual for the students will be downloaded. Similarly, explanatory videos link is there. When you will click there, complete video of each module they can see. So I can means it is a, you can say complete student need not to worry for other things, and completely they can write down their practical, they can see the videos, they can perform the practical, and all the modules, like it is not a single module, all the modules can be studied through the software. So this was, I was trying to show you how to use the software. And other than that, means, as you know, being a human being, our responsibility is also to prevent cruelty on animals. And it is a right tool to prevent cruelty on animals. Just 1 minute, I'm getting little connectivity issue. So, ma'am, actually, I am little bit their connection or some technical error is there. So this was means from my side, like what is virtual pharmacology, what are their implications, and I have tried to show you one module also, how to use the virtual pharmacology software. And in fact, in coming Indian Pharmacological Society conference at Chandigarh, we are having sessions as well. We are having our demonstration as well. Similarly, at Indian Pharmaceutical Congress, we are also associated with them also. There will be demonstration sessions. So we welcome you all on that IPS also, IPC also. And being a principal mentor of experimental pharmacology, XPharm, I thank the institution, the principal sir, and all learned faculty member for inviting me and giving the opportunity to students and means to faculties also to learn the application of AI and to refresh their knowledge. As I told in my start that studying the books is one part, but making themselves abreast with the latest technologies, they should always attend such type of FDPs. Then only they will get to know what newer things are coming in the teaching technologies and everywhere. So thank you. Please.
Thank you, sir, for such an insightful session. Now, to all the participants, if you are having any queries, you can directly ask question to our speaker, sir, or you can drop your question in the chat box. Any participants having any queries, you can directly ask to our speaker, sir, you can drop your message in the chat box.
Yeah, if any query is there, they can ask.
Anyone, any queries? Which all, a question from Dr. Ashmita Shah, which all softwares can be used for regular B.Pharm classes?
You can use this XPharm, experimental pharmacology. This is sufficient to cover all the B.Pharm modules because it has included the physiology modules, because in PCI syllabus few physiology modules are there. So we have already included that in the experimental pharmacology as additional features. So in case of BPharm, there is no need to go for any more software. XPharm is sufficient enough. Although one more full-fledged XPhysio software is there, but no need to go for complete XPhysio because that contains number of additional modules which are not needed. Only certain modules are required from XPhysio. So we have already included those modules in the XPharm module itself. So XPharm is sufficient. Experimental pharmacology XPharm.
Next question is from Sonam Bilwet. Virtual software free of cost, how to cite?
Means your institution has to subscribe the software and then you can access it one. It is having very nominal cost and institutional access. So institutional license has to be there. So you have to get the login panel login from your institution.
Thank you, sir.
Like that.
So now we will
Yeah.
proceed.
Means
So I would
it's
request,
although it's means
sir, you can continue.
yeah, I was saying that means to make it very easily accessible, it is available at a very low cost and it is in the form of institutional license. So you can simply, to whomsoever institution you are associated, you can contact to the pharmacology faculty and you can access the software. Like that. Yeah, please, ma'am. No.
Thank you, sir. Is anyone having any questions or doubt? Any participants having any doubts? So thank you, sir. Now we'll proceed. So now I would request Professor Dr. Subhasis Devnath, sir, Principal of Bharat Pharmaceutical Technology, to present the certificate virtually to our speaker.
Namaskar, sir.
Namaskar. Thanks for the invitation, sir.
Okay. Thank you very much, sir, for your beautiful presentation.
Thank you, sir.
And also, like, I just want to know, is there any scope, like you are going for updation of the software? Now, see, virtual reality also came.
Yeah. So
Which
we are
will
adding
make the
that
students more friendly.
Yeah, sir. You are correctly said. We are adding more and more AI tools and virtual reality options in the software so that student will get more realistic experience while performing. But we have to keep in mind that it should be simple also because if it will be very difficult, then again the technical issue will come and student will be away from learning. So we are matching both the things and we are coming with more and more features, sir.
Yeah. Thank you. Thank you that we are able to know this.
Yes.
And now I request Pralay sir to present the showcase the certificate. So thank you very much for your presentation and time, sir. And I request you to accept the certificate virtually.
Thank you very
And
much, sir.
hope we'll meet. Yeah, we will meet in the coming future someday in our institute or someday. Thank you again, sir, for your kind acceptation.
Thank you, sir. Now I would like to request Dr. Santosh Ganguly, sir, to deliver vote of thanks.
Thank you, madam. Hope I am audible.
Yes, sir, you are.
Thank you. A very good afternoon to all. It is my profound privilege to deliver the vote of thanks at the conclusion of today's highly enriching and thought-provoking session. On behalf of the organizing committee, I extend our deepest gratitude to Dr. Dharmendra Ahuja sir for delivering such a profound and enlightening lecture on the topic use of AI in pharmacology teaching through experimental pharmacology software. Sir, your clarity of thought, depth of knowledge, and ability to articulate complex ideas with such simplicity and precision have truly captivated us all. You have shown us how artificial intelligence is not merely a futuristic concept, but a present-day reality that is transforming the way pharmacology can be done. By illustrating how experimental pharmacology software can contemplate conventional teaching methods, you have provided a vision of how students can gain a more comprehensive, practical, and engaging understanding of the subject. Your emphasis on the integration of AI with pharmacological education is of great relevance, especially in today's time where the healthcare sector is continuously evolving. The way you highlighted the possibilities of creating virtual experiments, simulating drug effects, and enhancing students' engagement through technology was truly fascinating and inspiring as well. It has not only given us new ideas to think about, but also has motivated us to adapt and innovate in our own teaching and learning practices. I would also like to take the opportunity to sincerely thank our distinguished participants who have joined us today and contributed with their presence. Your questions and reflections have added immense value to the session, making it even more meaningful and engaging. As we conclude, I once again extend my deepest appreciation and gratitude to Dr. Dharmendra Ahuja sir for his valuable time, exploratory insights, and inspiring words. I am confident that the knowledge shared today will not only resonate with us, but also guide us in our future endeavors. Thank you all once again.
Thank you, sir. So here we are concluding our first session now. And the second session is going to start. Our second speaker has already joined. So we are extremely fortunate today to have with us an incredible speaker, Dr. Kunal Bhattacharya, Associate Professor, Department of Pharmaceutical Science, Pratiksha Institute of Pharmaceutical Science, Guwahati, Assam. So I request Ms. Pragya Datta, ma'am, Assistant Professor of Bharat Pharmaceutical Technology, kindly grace our speaker. So I request Ms. Pragya Datta, ma'am, to kindly grace our speaker.
Just 1 minute, madam. There might be some technical issue. Just 1 minute.
Okay, sir. Sorry for the inconvenience. There is some technical issue. Just wait for a few minutes.
Sorry for the inconvenience. Good afternoon to one and all present here. Today, I'm extremely honored and elated to get an opportunity to introduce one of the most accomplished speaker, Dr. Kunal Bhattacharya sir, to this e-faculty development program. Dr. Bhattacharya is a pharmaceutical chemist and academic whose career began as R&D scientist in Mylan Laboratories, where he gained expertise in analytical method development and validation. Building on this foundation, he transitioned into bioanalytical roles at Lotus Labs and later in academia, where now he serves as an associate professor of pharmaceutical chemistry. His research spans computational drug delivery, multi-epitope vaccine designing, bioinformatics, and neurodegenerative diseases with a PhD focused on Alzheimer's. Dr. Bhattacharya has authored numerous peer-reviewed publications, book chapters, and edited as CRC Press volume on neurodegeneration. He holds international patents in vaccine design and bioinformatics platforms targeting SARS and Covaxin 2 and Pumala virus. Recognized with fellowships from IASC, INSA, NASI, and CSIR, and awarded Best Researcher of the Assam Royal Global University by Scopus. He continues to mentor students and review for top journals, including Springer, Scopus, etc. His interdisciplinary journey from industry to academia reflects a commitment to innovation and excellence in pharmaceutical sciences. Thank you, sir. The stage is all yours. You can continue. Sir, please unmute
Sir, you
yourself.
are,
I think I'm audible now.
yes, sir, you are
Yes.
audible.
Okay. Thank you for the warm introduction, ma'am. I would like to first thank Principal Sir, BPT, and the management of BPT to provide me an opportunity to talk on this e-faculty development program. So today we'll talk on basically computer-aided drug design. Let me share my screen. Is the PPT visible?
Yes, sir, it's visible.
Okay. So my topic will be principles, tools, and applications in modern drug discovery. So the first thing is, what is CAD? After COVID, we have came across this term, and nowadays you can see that every pharmaceutical chemist or pharmacologist, they are all using this software for their PhD purpose and personal research work also. But when I started back 4 or 5 years, that time it was hardly known by few people only. And the thing is that it is not actually new. It has been there from 1960s, but currently it is on trend after the COVID, because most of the COVID drugs are also identified with the help of CAD. That's why we could see that so much fast we could develop drugs. Now the time is that every year we can find new viruses, new bacteria coming up, and we cannot give that much time that we have 10 to 15 years for development of a drug. By the time multiple viruses and bacteria will be there, and we need to counter it. So to make the process faster, now CAD is involved along with the latest AI and quantum computing, along with machine learning also. So why CAD matters in our modern drug discovery? As we know from traditional drug discovery method, it take almost 10 to 15 years to develop a drug. And also, while developing a drug, it takes a lot of cost for the pharmaceutical industry. Around, you can see $2.6 billion are taken for a drug. And mostly 90% of the experiments fail during the drug research, and only 1
to 2% of the complete research you will find available in the commercial market. So that is one factor that why branded drugs are also costly compared to the generic ones, because of the money they are spending during the research work. So that is one thing we need to tackle to make the medicines more affordable, so that if the research cost is less, finally what will happen is the available medicine in the market will also cost less. So after during COVID, we saw that the vaccines were generated within a year. So how it was possible? It was possible because of this kind of technologies which are available nowadays. That time, quantum computing was also not very much popular, but these days you can see AI agents are there, automation is there. Most of the thing in every day, I think you can see in newspaper that for some disease for which the drugs were not available 5 to 6 years back, now with the help of AI and machine learning, you can find precise target. Okay, so that is one thing. Then we can tackle high failure rates with the help of CAD. Then time is also reduced. With traditional method where we spend 10 to 15 years, but with CAD, it will take around 5 to 7 years due to the early stage acceleration of the drugs. Then if we see the evaluation of the CAD from 1960s, it started. Basically, that time the computational power was less. So what people used to do is that what we study in our B.Pharm syllabus in medicinal chemistry, SAR, structural activity relationships. So they used to validate those things using computers, but due to lack of graphical power and computational power, only a small part of that could be done during that time. But still, when back in that time, 10, 15 years or 20 years back, HIV was like a death sentence, that if someone gets HIV, the death is almost prominent. But due to the help of CAD, we discovered drugs like Secukinumab, because of which a person can lead almost a normal life nowadays with the help of HIV, if he is suffering from HIV also. So those things got possible because of this new technologies, where earlier, because of certain diseases, people used to die. Now people are living comfortable life with that. Here you can see some of the examples of different classes of drugs which were developed with the help of computer-aided drug design, starting from antivirals to antibacterials, cardiovascular drugs, and mostly anti-cancer drugs also. So you can see these all drugs are FDA approved, and it started from almost 1995, and the latest drug is FDA approved in 2011, that is anti-cancer drug. So still drugs are coming and FDA approvals are also coming. So in coming days, you will see that many of the diseases are which were non-treatable are maybe going to be fully treatable in coming time. So coming to the principle of CAD, the first principle, as we discussed, which started in 1960s also, that they used to see the structural activity relationships. So still today, the basic principle remains same as the structural activity relationship activity of 1960s. So you can see here as an example, when we are taking aspirin. So what is basically SAR? What we do is that we just modify the groups and see whether it is increasing the activity or decreasing the activity, whether it is increasing the toxicity or not. So different kind of modifications we do on the basic skeleton of a drug. So for an example, we have given here that if you are taking aspirin, so look at the IC50. It is almost 10 micromolar because of this acetyl group, which is crucial for the COX-1 selectivity. Now, when you remove it, the IC50 changes from 10 to 50, which is because of the missing acetyl group, which is actually important for the COX-1 selectivity. Then you come back to benzoic acid, then IC50 again increases, and further to methyl salicylate, it goes to 200. So it started from 10 micromolar and it is now going to 200. So that's how you can see that small groups, when you change, what will happen. So earlier, when we did not had computational power, we had to manually see, manually synthesize, and each and every structure we have to generate and confirm by mass spectroscopy, NMR, and then we have to do in vitro tests to identify this IC50 values and all those things. So it used to take lot of time and lot of expense, which is now has been almost eliminated by softwares which are available. By one click only, you can draw in ChemDraw. You can, as per your wish, you can change the group and you can check the IC50 values, toxicity, admit prediction, everything you can do over the computers. So it gives you an initial edge to develop the drugs. Then comes the another important point, that is pharmacophore elements. So these are the groups which are basically responsible for your activity in any kind of a drug. So the major elements which should have in a drug is the H bond donor, hydrogen bond acceptor. Then these two are usually required for forming the bond with the proteins. Strong bonds are formed when you have good number of hydrogens in that. Then you also need hydrophobic regions, aromatic rings, and because electrostatic environment needs to be generated, for that you need positive and negative charges also. So these are some of the important elements which is required in our drug. So while designing a drug, you need to consider these things. It should be there in your structure. And what are the challenges in this while you do this SAR related thing? That data quality is an important aspect. The need for high quality standardized data sets, because we are using most of the data sets from different sources in software. So your data should be highly standardized. Then you are using machine learning models nowadays. So machine learning models, how they are interpreting and how the predictions are made, how accurate that is. So that needs to be also seen. So nowadays you might have seen that everyone is using Autodock and docking, molecular docking. If you talk to any person who is doing your computer-aided drug design, so the only thing they have in their mind is molecular docking. That is having a software. What it does is that you give a protein and a ligand, and it will interact in the software, and it will give you a binding affinity. So anything you give to that software, it will definitely give you a binding affinity, whether it is less or more, that doesn't matter. What matters is that how accurate is that, because most 90% of the cases, if you see, if you take the docking complexes to MD simulation or free energy analysis, 90% of them will fail. So people need to understand that molecular docking is not the only part in CAD. That is a very limited part in CAD, and CAD is a vast thing. And there are multiple steps of validations when you start first process in that pipeline. So you need to understand all those aspects while designing the drugs. Next, you have two choices how you can design a drug. If there are two possible scenarios, you may have the structure of the protein for which you want to design the drug. And the next possibility is that the structure is not available, but you have experimental, what you can say, experimental data that some of the ligands or drugs binds to this particular protein. So what you do is that in CAD, there are two principles: structure-based drug design and ligand-based drug design. You are going for structure-based drug design when you have the proper structure of the receptor. So if you have the receptor, what you need to know is that where your ligand will bind, you need to find out the pocket, the active pocket where your drug will bind, particularly to inhibit or modulate the protein's activity. So that is a simple thing. You have the structure, you just find the ligand and you validate using MD simulation, or you can do pharmacophore modeling to improve your existing drug. And then you have another principle, that is ligand-based drug design, where you have drugs which are known that it inhibits a particular disease, but you don't know that what is the structure of that protein where it is interacting. So you design different kinds of ligands through pharmacophore modeling by seeing the existing drug which is available in the market. There are multiple structures you can generate which might have similar properties to your existing drugs. So that will come under the ligand-based drug design. And ultimately, for a proper drug design, you are combining both of them. So you can see the workflow here for ligand-based drug design. So you need to collect the known actives, then you do the QSR modeling, then pharmacophore generations, and finally you screen it. So you have known active molecules, but the drug target is unknown. The structure of the protein is unknown. Now for this, we have in these days, you can find different softwares also like AlphaFold.
Sorry, sir, for the inconvenience. You are not audible. No. Sorry for the inconvenience. Our speaker, sir, is having some technical issue.
Shantanu sir, if possible, call once.
Yes, sir.
Kunal sir has joined the meeting, so we are going to continue the session.
Sorry for the interruption because of some.
Sir, you are not audible actually.
Am I audible now?
Yes, sir, you are audible.
Okay. Okay, so sorry for the interruption because of network error. I was talking about the structure-based drug design. So you have the workflow here, which starts from the target structure. You need to find out the target structure based on, we'll see that how we can fix the target structure based on X-ray, NMR, and homology modeling in the practical session. So first you need to find out a target against which you will test your drugs. Then you need to find out the pocket where your drug will bind. Then you perform the molecular docking and it is validated by your MD simulation. So in this case, you have a known structure. The protein structure is known and the design process is also simpler compared to your ligand-based structure. So there are 4 key points which you need to consider during this structure-based and ligand-based design. First one is QSR modeling, as we discussed that 2 types of QSR there, 2D and 3D QSR, and you predict the structural activity relationships based on changing different groups and moieties present in the structure. Then you validate those by seeing the binding affinity with the help of molecular docking. And if required, you can modify your structures using pharmacophore modeling. And finally, you can validate your molecular docking again with the help of molecular dynamics. There is one more validation step you can add, is that free energy binding analysis that I have not shown here, but that can be also added. And then after, when you have completed this process, then you need target identification and validation. While you are identifying the target, what is a target basically? Any kind of proteins or genes against which you are testing your drug is a target, right? Then you need to validate your target with the help of either experimental confirmation in vitro or in vivo, whatever you prefer. And after that, you need to do the target prioritization based on ranking the target based on the therapeutic potential and novelty. And finally, you do the druggability assessment. So while you are finding your target, what you can do is that there are multiple approaches with the help of which you can find your targets. You can use genomics and transcriptomics. Then you can use proteomics and interactomics. Interactomics is like how 2 different proteins will act together, how they will connect to each other. And similar kind of things can be done with the network pharmacology. So all these are different branches of bioinformatics and it will require additional workshops to tell about these things individually. So then also you can do the literature and database mining, that if you are studying about a disease, first you need to understand that what kind of drug targets like proteins or what kind of genes are involved in it. Then you need to understand about the available drugs which are present. If you want to modify certain kind of things, you can check by using ChemDraw, draw certain structures and use them for molecular docking. You compare the binding affinity and validate using the MD simulation process. Then for target validation methods, you can have further genetic, pharmacological, bioinformatics, and clinical evidence. Then finally, once you have found that, yes, your target is fixed, your ligand or drug is fixed, they are binding properly to each other, they have good binding affinity, then what comes next? The next is the ADMET, that is absorption, distribution, metabolism, excretion, and toxicity. So if the drug fails in any of that, so it needs to be discarded. That's why most of the drugs, when they are tested, they show good activity, but one of this factor fails during the trial. That's why 90% of the drug fails during the research. So for a good drug to come into market, it should have all these parameters fixed. It should have good absorption, it should be distributed properly in your body, then metabolism should be good. Then you need to see that whether it is excreting very fast or very slow, whether it is showing any kind of toxicity. So we all know that there were different cases of toxicity. The drug came in market, and after that also, that after phase 4 also, it was found that so many drugs showed different kind of toxicity. So classical toxicity cases are already present. So recently, if you see I forgot the name of the drug, but it was used for reducing the weight. So it is now seen that it is showing different kind of toxicities, and cases are filed against that drug in US courts. So these things happen. So your drug, not only it should have good activity, but it should have all these parameters fixed so that it doesn't harm you. So another parameter is drug likeness, which is measured using CAD. So how you fix this ADMET? You have 5 rules, which are called as Lipinski rule of 5. If a drug is is falling under this category of Lipinski rule of 5, and it is passing all these rules. So we can consider that that drug is safe and we can take it to further study. So
Sorry for the inconvenience. Our speaker sir is having some network error.
Sorry for the inconvenience. Yes. Sir, start. It will take 1 to 2 minutes. Sir is joining actually.
All the participants, please wait for a few minutes. Our speaker sir joined the meeting, so he was starting.
very sorry, because of the weather conditions, the network is going repeatedly. Anyways. Okay. I think it's visible now again.
Yes, sir, it's
Yes,
visible.
sir.
So I was talking about drug likeness scoring. So there are, apart from the drug likeness score, there are others rules also from different scientists like Weber's and others. So basically we are following the Lipinski rule of 5 only. Any of the drug, if it is satisfying all the 5 rules of Lipinski, we can consider that we can go ahead with those drugs. If 1 or 2 violations are there, based on your judgment and your expertise, you can consider those drugs also. Like first one, if I give you an example, that is molecular weight, it should be less than 500 Daltons. So if you are researching about brain related targets, then obviously it needs to be less than 500 Daltons. But if you are using for any skin related or any kind of other superficial thing, it is not mandatory that it should have less than 500 Dalton molecular weight. So based on your research interest and your experience, you can play with these rules, whatever is there. So why it matters? Because through this in silico admit predictions, you can not only predict that which rules, which drugs will pass the Lipinski rule of 5, you can also see that which drugs might cross your blood-brain barrier. That option is also there. Then you can see that if some drugs are being taken, whether it will interact with your cytochrome P450 enzymes or not. So if it is interacting, that means there are certain drugs which if you are taking together with your investigational drug, that might interact and activity might change for both of them. So you need to also consider that whether it is interacting with the cytochrome enzymes or not. So all these things you can check through in silico studies, and later you can confirm with the help of in vivo or in vitro studies also. So that gives you an edge, and your drug discovery time is also reduced to an extent. So some of the tools which are basically used for CAD is for molecular docking. You can see, I think Autodock, Veena, and Pyrex, most of you have used. And for good institutions which have enough money to buy Schrodinger, then Schrodinger Glide is a very good option. You can do multiple things in Schrodinger Glide. Then for virtual screening, again, you can use Pyrex also. For database, you can use Zinc, Kebi, and all these databases you can find where multiple structures, thousands of structures are free. And there are certain databases like Zinc, where if you find some structures which are showing good binding affinity, you can directly buy from them, and you can test those drugs against your in vitro models also. Then for pharmacophore modeling, again, you can use the Glide software and Discovery Studio and Pharmate also. Then for your ADMET prediction, you can use Swiss ADME, PKCSM, and molecular dynamics. We have Amber also, then Gromax, Desmond. Gromax is free version, so anyone can use if they have a good system. For molecular dynamics, you need good graphical power. So for that, you need a better system to check all these things. Network pharmacology is one of the aspects where you can find actual mechanism how a drug is acting. So in network pharmacology, what is happening that when we are doing experimental things, we can see that yes, a particular drug is showing any COX activity, or sorry, any kind of activity, anti-cancer or antiviral activity you found through in vitro or in vivo. But through animal studies also, you cannot understand properly that what is the mechanism behind it. So to understand the mechanism, you can use network pharmacology or systems biology. You can perfectly know that which of the protein targets can be targeted. And earlier the concept was like, for a particular disease, only one target were chosen. But nowadays, what is there? It is called polypharmacology. So you have multiple targets associated with a particular disease. So not only if you target only one, then nothing will happen. In case of cancer, there are multiple pathways which are involved in a particular cancer. So if you just inhibit a particular pathway, it will not be sufficient. So you need to target all those together. So now the newer concept is that your one drug should target multiple pathways. Earlier it was one drug, one pathway, or one target. So now it is one drug but multiple targets. So that you need to keep in your mind while designing the drugs. Then you have AI and ML in CAD nowadays. AlphaFold, which I told you, that earlier the problem was we could not determine the structure of proteins. So what you can do is that you can give the genetic sequence of the proteins nowadays to AlphaFold, and it will create the structures, and then you can test the drugs against it. So everything is possible. Now machine learning is coming up, quantum computing is coming up. So all those things will create newer drugs in coming 5 years. You can see whatever drug development you have seen in last 15 to 20 years, you are going to see more than that in coming 5 years. So our generation will see those things which the world has never seen actually. Nowadays, you have also seen, I think, CRISPR-Cas technology, that is gene editing. So now you can treat those kind of diseases which were not treatable just by editing your genetic sequence. You just remove out the gene causing your cancer and you will be cancer free. That much it is simple. But for like coming to common people, that will take little bit time. So what are the current challenges actually which we are facing? Like I said that every student, every teacher, or every academician is doing molecular docking. But how far it is actually proper? How every docking will give you a score, but how accurate that is? Have you validated the software? Have you validated your results? No one is doing that. Everyone is just taking that binding affinity and trying to publish it. That is not actually what CAD suggests. So that is one of the problem which we are facing. Then you have protein flexibility, because docking, what we do is that in that time the protein is in static formation, that is not moving. But actually, if you see inside your body, the proteins are not at rest. They are folding, they are uh, becoming compact, they are becoming flexible. So all these things are happening inside your body. So that needs to be validated, and that is done by molecular dynamic simulation. Then you have allosteric site prediction, then membrane protein structures, protein-protein interactions. All these things are happening, but to a limited stage. So these things needs to be validated. As I said, quantum computing is coming. So these problems will solve over next 1 or 2 years. Then you have your biological challenges like disease heterogeneity, target validation in vivo. Sometimes, most of the times you see that you are getting good docking results, but in vivo, it is completely negative. So these problems are there. Then resistance mechanism is there. Last time you have seen during the COVID that vaccines are coming, but different variants of the virus are coming after 1 or 2 years, and your vaccines are failing. So in that case, what you can do, you can take out the genetic sequence of patients infected by different variants of the virus, and you can take epitopes and join them together to make virus resistant vaccines also. So that's how multiple things you can do. And what are the emerging technologies apart from your regular, what we see, molecular docking and simulation, all these things? Next, quantum computing is coming, which will solve most of the complex molecular interactions. Then cryo-EM revolution is there. So what cryo-EM does is that it predicts the structure. It is kind of a microscope. So you can find or predict accurately what is the structure of a protein. So that is also developing. Then you are having graph neural networks, which shows better molecular representations. Then collaborative drug discoveries are happening. Then nowadays, you can see that some of the companies are there in US who are validating their drugs in virtual patient models, which are called as digital twins. So a time will also come that when clinical trials will also be reduced to digital. Okay. So what is our future outlook for this CAD related thing? In coming years, we might see 70% reduction in discovery time by 2030 and 5x increase in the success rate of drug research. And for each drug, we might save almost $1 billion. So that is what CAD gives us. And we are expecting it to be better in coming days. So I will stop here for the theoretical part, and maybe we'll move to practical. I will show you a little bit that how properly you can do molecular docking. So if you are okay, we can proceed with that now.
Yes, sir, you can proceed.
So I'll take a small, as we are talking about antivirals, so I will take a small example of antivirals. So for the first thing, what you need is your protein target, right? So there are multiple databases by which you can find your protein targets. One of them is called RCSB, Protein Data Bank. So if you type RCSB in Google, so you will find this site, RCSB. So here, whatever structure you want will be available. Scientists across the world, when they solve the genetic sequence, they put the structure in this RCSB data bank. Okay. So suppose in this window, you can see there is a structure of the COVID-19 main protease in complex with an inhibitor N3. So this inhibitor N3 is an investigational drug, which they have found experimentally that this N3 drug is already inhibiting this COVID-19 protease structures. So when you search here in this search panel, anything you search like spike protein. So you'll get multiple structures which are deposited by different scientists. You can see the name here, Takeda S, Yamashita. Then I think mostly this is from Japan. Then you have 3QRS. So every structure will be given a particular code, which is shown here, like 3AQJ, then 3QR7. So these are the codes which are available for each and every structure. So there are multiple things which you need to take care of while downloading a structure from RCSB site. Another popular site is your UniProt. Just type UniProt over Google and you will find this site. So it is similar to your RCSB. You can type here also spike protein. Any protein if you want. If you want related to inflammatory diseases, you can take COX1. So it will give you an option that how you want to see the results, whether in form of cards or table. That is based on your preference. Whatever you want, you can select. So as I have typed COX1, you can see that cytochrome oxidase subunit is coming. Then cytochrome oxidase subunit for different type of structures are coming. So what you need to see is that if you are looking for human related structure, in the left panel, you can see that popular organisms, human. So you need to click over human and you will see all the structures which are related to humans only. Okay. So any structure, mostly the first one you can take. Suppose this one. So you will get all the informations in the left panel. Like if you directly want to go to the structure, just click here in the structure and it will show you the 3D structure of that particular protein target. And you can see here, this is AlphaFold structure. That means it is not a chemically or experimentally validated structure. It is determined with the help of Google's AlphaFold structure, because they did not find the structure for this. So every structure, suppose we take the spike protein here, in UniProt, you will find along with the experimental structure for every protein target, you will find the AlphaFold structure also. There are multiple informations of this structure, family, domain, sequence, everything you can find whatever is required for you. Like this structure you see, this all whatever is written PDB are all experimentally determined structures. Okay. So at the end, you might find AlphaFold also. I think there are multiple structures for this. Okay, if you go to the end of the structure, you will find AlphaFold also, which is present here. So what you need to see is that this is the CB0B. This is your PDB ID. If you put this identification number in your RCSB also, you will find the same structure. So you will have multiple options to download here. There are multiple databases from which you can download this structure. One of this database is RCSB. So if you click here directly, RCSB site will open, and from there you can also download this structure. So for today's example, we are taking the protease structure of SARS-CoV-19 6. 6LU7. So this is the identification number of your main protease of COVID-19, that is 6LU7. So what are the informations which are given here? See here, it is given classification. It is a viral protein. Then organism, severe acute respiratory syndrome, coronavirus 2. So if it is related to human, here it will be written Homo sapiens. Instead of this SARS, it will be written Homo sapiens. So you need to take care of that also, because what happens is that a particular protein can be present in different animals. It can be present in humans also, it can be present in animals also. So based on your research requirement, you need to first see what is the organism given here. If it is Homo sapiens, then only you need to take the structure. Then expression system, that means where it was cultivated mostly. So it was expression system for this is E. coli. Mutations are no. If you want to see your drug against mutated structures, then you can see that also. But generally, what we do is that we see the normal structures and the mutation here should be no. So then you see method of experimental data snapshot. Here it is written X-ray diffraction. So always choose X-ray diffraction structures over NMR structures, because if you see NMR structures, you will find many of the chains are missing or amino acids are missing from the structures. That will not give you accurate results when you do molecular docking or dynamics. So you need to choose X-ray diffraction as your preferred method. Then resolution. Resolution means it is measured in angstroms. So as much it will be less, that much is better. You preferably take less than 2 angstroms, but if the structure is good as per your expertise, then around 2.5, 2.16, 2.2, these are all okay. So here we can find that 2.16 angstrom is there as a resolution. So we can take this for our experimental testing. And here in the PDB validation, you can see there are certain percentile ranks, and you can see red mark and blue mark here. So these ranks, if they are mostly in the blue side, that will give you a better PDB structure. So find those kind of structures which are having this property, that it should be organism should be Homo sapien, mutation should be no. If you don't want to investigate a mutated protein, then it shall be no. Then method should be X-ray diffraction. Resolution should be less than 2 angstrom. So this certain few parameters you look before you download the structure. So if you want to see proper information about the structure, that how it was developed, then you can find this paper also. The paper has been given here. If you can Google it, you will find the paper about this structure. When you come down, you see the information about the chains which are present in this structure. You can see the molecule is 3C-like proteinase, that is the protein structure. It contains only one chain. Then there are proteins which might have multiple chains also. You can see A, comma, B, comma, C. So you might have multiple chain proteins also, that also you can take. Then comes the sequence length. It is showing 306. That shows that how many amino acids are present in your protein structure. So then you come back to the lower section and you will find that the ligand which is attached to it, what is the molecule's IUPAC name. So you will find it here. What we were talking here was they have named it as inhibitor N3. So what is inhibitor N3 actually? Inhibitor N3 is nothing but this molecule. Here the structure is also given. So these are some of the important things which you need to see before downloading the structure. So for downloading the structure, what you need to do is that go to this download file and you select this legacy PDB format. Okay, so I have already downloaded it. So just if you click it, it will download in this format, .pdb format. So your target is now fixed, that you want to target the 6LU7, that is COVID-19 main protease. Okay. Now, suppose we have seen that during COVID, against the COVID virus, remdesivir was extensively tested. So you can find different drugs also from PubChem database. So if you type PubChem, so we'll get this window of PubChem. Any drug you want, any 2D structure or 3D structure of any kind of drug available in the market, you can find here. Just go over it and type remdesivir. You can check any drug. So like this kind of result you will come up with, and select this remdesivir here. Then you will have n number of information around this structure. If you see the content list, you from the structure to toxicity, pharmacology, then from where you can bind, where you can buy chemical vendors, and any related records related to this structure you can find over this site. So both 2D and 3D structures as well as the crystal structure developed by the CryoEM will be available here. What you need to download is the 3D structure from here. So first they are giving the 2D structure here. Then if you come below, you will find the 3D structure here. Just come here in this 1.2 section, 3D conformer, and download coordinates. Downloads this SDF file. There are multiple formats, so you need to download the SDF format. So once you download, you will find this kind of conformer 3D compound. So now both our drug as well as the target is fixed, right? Just delete those other things. So we have these two files here. So our target is fixed, our ligand is fixed. Now we need some softwares to analyze it, right? So first software which I recommend is your PyMol. Just type PyMol over Google and select this link, educational use only. Okay, select this link and you need to fill out this form. So once you fill out, you will be given an educational license which you can use for downloading the PyMol. So PyMol you can download from here, from the first link, and you can take the educational. Here you can download for Mac OS, Linux, Windows, whatever your system is, and you can take the educational license from this link. Okay. So once you have downloaded PyMol, the second software which you will require is Discovery Studio. So for this, you need a institutional email ID. If you don't have, you can take from anyone and just download the file and you can share with anyone. So for this, you need a professional institutional email ID. By filling this email ID and the form, you will be able to download the Discovery Studio also. Next, what you can download is Chimera. So write Chimera here, and the first link comes is download UCSF Chimera. So once you click it, you can do multiple clicks as per the instruction and download this Chimera 1.19 Win64.exe, which is for the Microsoft Windows. If you have Mac OS or Linux, you can download this other two. Then one more is required, that is called OpenBabel. So type OpenBabel here, and the first link you can click from SourceForge, that is OpenBabel, and directly download from here. It is just a 21 MB file, so it will not take much time also. So these are the tools for your visualization purpose. Next, you need your main softwares, like with which you will be performing your docking. So multiple types of docking you can perform. You can do virtual screening, you can do one-to-one docking. So multiple ways are there, and multiple steps or ways in different softwares are also there. So whatever you prefer, based on that, you can select any one of them. So for docking, what you need is first software, which is very famous, that is Autodock. So if you just write Autodock here, you will find the first link, Autodock. Download Autodock 4. When you come to this page, in the download section, you will find the exe file for different OS systems. Mostly you are using Windows, so you will be downloading Autodock for Windows. So once you download Autodock for Windows, another tool you need to download, that is MGL Tools. So next thing you need to download is this MGL Tools here in the download section. Again, download for MGL Tools Win32 1.5.7. So these two tools are required for your docking. And if you want to do virtual screening, you can also download Pyrex. So Pyrex is in background, Autodoc is only running. There are two algorithms of Autodoc, Autodoc 4 and Autodoc Vina. So both are inside this Pyrex. So you can use this free version, 0.8 version. If you have academic opportunity, then you can buy this academic license also. Otherwise, you can use this free version. So nothing, just you need to download this and click by click, it will install. So in this system, I have already installed all these things for showing it to you. So now we have all our softwares also, and we have our target and Lichen. So once you have downloaded the softwares, there is a particular step I want to tell you while you install. When you are installing the Autodoc file, what you need to do is that I'll just show you once. It is there. Like this kind of file you will get, Autodoc suite. So when you install it like this, so destination file, it will ask that where you want to install. Browse it and go to your main folder, that is this PC, your C drive, and make a new folder. Make a new folder like this and name whatever, maybe Autodoc, but it should not have gaps like Autodoc space docking, like that. It should not be there. It should not have any gaps. Okay, any single name you can write and save. Here for this purpose, I have already made ADT folder. So you can use ADT folder or Autodoc folder, but there should not be gaps in the name, and install particularly in this folder only. Whatever it will be given by default, you just remove it and install in that folder. So once you have installed it, then you install the MGL tools also in the same folder. Like this MGL tools you will find. So you install MGL tools also in the same folder. Browse it, go to C and select ADT where you have already installed your Autodoc 4. Okay. So once your softwares are ready, then you are ready for docking. So we'll open the Autodoc first. You just type Autodoc here, it will open up with the GUI of MGL tools. MGL means Molecular Graphics Laboratory, that gives Autodoc the GUI to perform the docking. So this kind of setup you will find once you open the Autodoc structure. So you have your, minimize these things, you have your protein structure for your easy identification. I'll just rename it to protein or receptor. And this one we can rename as ligand, which is your drug. So first thing when you download a receptor is that you need to see whether is there any missing amino acids or chain breakage in the structure or not. If it is there, the docking will definitely happen, but the score also you will get. But when you do the further validation steps like that, okay, so how can you see that? Just open it in Pymol, right click it and open in Pymol. So once you loaded the Pymol here, the license, how I told you to download, just browse and place your license here. For the coronavirus, and how you want to see that if there is any missing chain residues or not. See in the right bottom, you can see an option called a sequence. Just click it and it will give you the individual identification of the amino acids. This alphabets represents a particular amino acid. Okay. So this is the identification way how PyMol shows it. So these are nothing but amino acids which are present. So green ones are amino acids. Then you might have certain metal atoms or ions also present in the structure, which are given here like 02, ABL, PGE. All this kind of things will be your extra structures. Whenever you find single alphabets like this, then that means it is an amino acid residue. See here, if I start from ones, if I start looking from 1 to, if I scroll to the right side up to 306, that means it is having 306 amino acids in total. So there is no breakage. There is no missing amino acid here. There is no gap. It is completely filled. If there is gap, that means the structure is broken somewhere. Okay. So before docking, you need to prepare the structure. So what do I mean by preparation? Preparation means for docking, I need only the plain receptor structure, no water molecule, no ions, nothing. Okay. If I am not doing ion docking or metal docking, that is a different aspect. For general rules, we only need the protein structures, and apart from that, everything needs to be deleted. So how you can delete? Just select this first and hold your shift button and select the last. So it will select all these things here present in the structure and right click it and remove. Now you can see only your amino acids are present here. Rest everything has been removed from the structure. Okay, you can rotate it here and see. There is in the visually also you can inspect that there is no breakage in the chains. Everything is complete here. So you can see three type of structures here, your regular beta sheets, helix, and your threads also. Okay. Then you just save it, export structure, export the molecule, and that will be in form of PDB, and save, and go to your original Autodock folder where you are performing the docking, and save as type. By default, it will be coming PDBX. So you should replace it with PDB only. Okay. Then give it a file name. Let me give it receptor intermediate. Okay. Receptor intermediate, I will give it as a file name. So for this part, we will close the PyMol now. Now, what we need to do with the receptor intermediate structure? This structure, we had already downloaded Chimera. So we will prepare it for docking. We will add certain charges to it and hydrogens to it, because hydrogen will provide the hydrogen bonding with the ligand, and charges will provide the electrostatic environment to it. Because in docking, what we are seeing is binding affinity, and binding affinity is mostly responsible because of the electrostatic potential, which is generating certain kind of charges. So charges, when there will be present, it will either attract or repel the atoms which are present in the system. So for that, we need charges also. So we'll just open it in the Chimera. Sorry. We had already installed Chimera. So we'll open up the Chimera, and from file section, we can open the structure. Receptor intermediate, double click, it will open. So this kind of structure will open, 3D structure. So what you need to do is just go to tools here in the panel, structure editing, and doc prep. Only uncheck this write mol2 file, because we need PDB file. So we are not interested in mol2. Other than that, everything default settings will be there. Add hydrogens, add charges. If there is incomplete side chains, that will be replaced. So these things are required. Just click OK. And whatever is coming, let it be like that default. Okay. And then you have another option here. It is putting the charge. That means it is asking you whether this amber is a force field. So it is asking whether you need to put AM1 residues or gas tiger charges. So gas tiger charges, basically you are adding for protein structures, sorry, ligand structures. So here, as we are adding the charges to protein, so we will choose the amber charges only. Okay. So AM1, let it be as default, and okay. So it's done. From here, doc prep is done, and save PDB from file. So here there is option to give the name. Just write receptor final. So that is our final structure for docking. So our receptor is ready for docking. So now we can open up the AutoDock. Okay, there is one more option I want to show you if you are interested. Chem3D also you can download. So like we have minimized the structure of your protein. For structure minimization, there is one more software called as Swiss PDB. So that also you can download and minimize the receptor. But for this session, I'm not doing that. And let's go directly to ligand structure. We'll open up the AutoDock. Okay, AutoDock is already opened here. So the first thing what you need to do is that you set the folder. Okay. Before that also one more step should be there. So when you have installed the AutoDock, if you go to the main C folder, ADT folder, you will find an folder called as 4.2.6. That is the AutoDock version. Just go inside it and you will find two files, AutoDock 4.exe and AutoGrid 4.exe. So copy both this file, and wherever you are putting your docking files, you copy and paste them. Okay. So that's how your folder is ready. Now you need to instruct your docking to choose this path only. For what you need to do is, from file, you go to preference and set. So here already my path is set to that folder only. If yours is not set, what you can do is that just come back to this folder where your requirements are there, and click on the panel. Click right side, you will get the path. Just copy it from here. Sorry, this is not my path. Okay, AutoDock. So just copy it from here and paste it in the startup directory. Paste it, set. Already mine is selected, that's why it is not taking. So set and make default. So that will be your option before you start the docking. Okay. Now everything is set. Now you can start the docking. Just go to file, read molecule. From the read molecule option, you need to select your final receptor file, which we have already prepared. So you select this, open up, and you will get the structure of your receptor. You can rotate and see if you want. So the next step, what you need to do is go to edit, and if there is any water molecule, okay, so if there is any water molecule, we have already deleted it with the help of Pymol. If you don't want to do with Pymol directly also, you can perform using the AutoDock. So what you can do is go to edit, delete, and delete water molecules. Okay. So, and suppose particularly you want to delete some particular amino acid from the receptor. So from the left panel, if you click this receptor final, the chain A will open up. We had seen in the RCSP side that it contains only chain A. So it will show A. So you need to again enlarge it by clicking the plus sign. So you'll get all the amino acids which are present in this structure, and come below. At the end, if there is water molecule, it will be there in the end. So how you can delete? Suppose I want to delete this amino acid, ILE281. Just click Here it will become yellow. Go to edit and delete the selected atom. So that's how you can delete any particular atom from the receptor also. So in our case, the receptor is already prepared, so we are not going to do anything from here. We will minimize it. Okay. So for our next part, what we need to do, we need to add the hydrogens. As we told that hydrogens are required. From here also you can add the hydrogens. So go to hydrogens here and add. Okay, before that, one more thing you can do is that you can check whether any missing atoms are there in the structure or not. So if you go to edit and click this MISC, miscellaneous, then you can check for missing atoms and select all residues. So it is showing that no residues missing atoms. That means my structure is complete, which I had already seen in the bimol structure. So from here, I can also confirm whether my structure is having missing residues or not. If it is having, suppose it shows that it is having some missing residues, then what you can do? Again, go back to edit, miscellaneous, and repair the missing atoms. So you can repair the missing atoms also from here on. So these two options are also there. If your structure is having problems, you can solve it from here also. Then what you can do is add the hydrogens. So go to edit, hydrogens, and add. So you have two options here, all hydrogens and polar only. You need to select the polar only option, because polar hydrogens are the ones which will connect to the heteroatoms, which will give you the strong hydrogen bonds. If you select all hydrogens, so duplicate hydrogens will be generated, which will connect to carbon atoms also, which is not required. So your computational power requirement will be more. So you choose only the polar only. So hydrogens has been added. Next, what we need to add? We need to add the charges. Go to charges and add compute Gastiger charges. So it will show that total Gastiger charges added is minus 3.0217. So it has added the charges. Next, what you need to see whether the charges are properly distributed over the receptor surface or not. So you again go back to charges and select check totals on residue. So it is showing that there is some problem. It is not evenly distributed. So click this spread charge deficit over all atoms in residue. Just click it and dismiss and check whether it has been properly handled or not. Again, go back to edit, charges, and check totals on residue. This time it is showing that no residues with non-integral charges found. That means it has been now evenly distributed across. Okay. So this much has been done. So after that, what you need to do is that go back to your panel. You have already deleted the water. You have already added the hydrogen. You have added the charges. Now go to this ligand section. Come to this ligand section. You have already prepared your ligand and open. So there is one problem here. See here, it is written files of type PDBQT. But what we have here, if we go to this folder, Autodoc, we have ligand.sdf file. But the software is asking you to give in the format of PDBQT. So if you remember, we have downloaded already the OpenBabel software. Open this OpenBabel GUI. Okay. Now here in the input format, there are multiple formats here. So you need to choose SDF, MDL, MOL format, which is your format of input file. Select here and browse to the folder where a ligand is present and select the ligand. Open up. It has opened the ligand. And next, if you go to this right side of the panel, what you will see is that it is already selected as PDBQT. There might be other selections. So you need to come back here to PDBQT, Autodoc PDBQT format. Select this and just click OK. Before that, output file is also there. Where you want the output file, give the same folder and provide a name, maybe ligand dot pdb qt format. So I have given and just press convert. It has already converted the SDF format to pdb qt. Let's check it. Okay, we have got the ligand.pdb qt format. Now come back to your Autodoc file and come back to ligand input, open, and now it is showing the ligand which is present in the pdb qt format. Now select this pdb qt format and open. So this kind of information about the ligand will be there. Just press OK, it is fine. And then again go to ligand, torsion tree, and detect root. It will detect the center point of the ligand. So that has been done. Once it has been done, what you need to do is that go to grid. Okay. Now choose macromolecule from here and choose. Now you have your receptor final, which we had already prepared. It is showing receptor final. You select it. It will show you an information. Press OK. And now receptor final dot pdbqt. Receptor, it will automatically convert in pdbqt format. So for docking purpose, what you need is that you need both the receptor and your ligand file in pdbqt format. So we have already converted ligand. Now it is converting the receptor. Save it. So receptor is also now saved in the format of pdbqt. Now go to again grid and set maps type and choose the ligand. Now choose the ligand and select ligand. So your work is done. Now computer has understood that what is your receptor, what is your ligand. Now you need to give the instruction that where this ligand needs to be docked inside the protein. In which part of the protein? See here the ligand is showing. In the screen, it is like looking far away from the receptor, but when binding will be there, you will see that it has bind to this receptor. So this is the complete structure of the receptor. Suppose you want to give instruction to the computer that bind to a particular site of this receptor. So how you can identify? Suppose you know from the literature survey or any active pocket screening software that a particular set of amino acids are active in this region of the receptor. So how you want to identify? If you want to identify the particular amino acids by looking at the structures, we cannot understand anything. Go back to the left panel. In the chain A, suppose you want that you want to see glycine, where glycine is present. Just click here, glycine. See here, this glycine is indicated by few of the yellow crosses. And like that, you can select other amino acids. So see, now if you have the information that around which amino acids you want to dock, you can just select this and you will come to know where these amino acids are present in this receptor. There is a second way that you will let the computer to decide that where the ligand will bind. That is called blind docking. So what we will do, we will not give any instruction. Today we will perform the blind docking only. So we will not give any instruction to the computer that particularly where to bind. We will let the computer to decide that where the ligand needs to be bound. That is called blind docking. So what we will do is that we will go to grid, then grid box. And from here, you can adjust the grid size. Okay. You can increase or decrease the XYZ coordinates. So we will adjust the coordinate in such a way. I'm showing you in a little bit hurry because of time constraint. So basic idea is that for blind docking, we need to cover the whole protein inside this box. That means I am telling the computer that you cover the whole protein inside your grid box, and where you find multiple locations and dock in multiple locations, the computer, what you will do, it will dock the ligand in multiple locations by itself, and it will give you the best pose where the binding affinity is minimum. Okay. So computer will decide the position of docking. So we have covered the whole protein. You can just see from any of the angle. Okay. Most of the protein has been covered. So next, what you need to do, go to file and close saving current. Again, go to this grid. Now we need to generate a document where the grid parameters are present. The XYZ coordinates are present to give instruction to the computer that we are doing blind docking and you need to perform the docking according to these parameters. So once you have set the grid box, what you need to do is that go to your this thing grid box and output save GPF. Okay. So you save the file in your particular folder where you have kept everything. We will just give 1.gpf. We will give this name as 1.gpf. Okay. So our grid formation is ready. Now, if you remember, I had told you to paste these two files here, auto.4.exe and autogrid4.exe. Now we need to run this autogrid4.exe. The parameter file we have made ready, and we will give instruction to the computer to use this parameter file to set a grid around the receptor. So we'll use that. We'll go to your docking, run autogrid. And here, see, in program path name, it is already given autogrid, but you need to choose the exe file. So we'll browse and select autogrid4.exe. And then you need to select your GPF file, which we had downloaded, and launch it. So this kind of window will come. It will be for a few seconds and automatically it will vanish. That means the parameterization is complete and we can proceed with the next step. So it is now gone. That means it is complete. Now we will proceed towards actual docking. So again, we'll go to run. Then we will go to we'll go to docking. We'll go to macromolecules and set rigid file name. Now docking can be of also two types. It can be the rigid docking or flexible docking. So in rigid docking, what happens is that your protein structure, your ligand remains rigid. Okay. But in your flexible docking, your structure is flexible. It can move in and around to adjust the poses. So for today, we are doing rigid docking. So we'll select the macromolecule, then rigid file name, then we'll choose the receptor final.pdb qt. Open this. So we have already opened, then go to again docking and ligand. Already we have given the information of our receptor. Now we will go to ligand and choose, and we will choose the ligand and select ligand. Accept it. The work is done. Now go to docking. Now docking needs certain kind of algorithms. There are multiple algorithms which with the help of which you can do docking. So the mostly used is genetic algorithm. So we'll search parameter here. We'll go to generic algorithm, and here you need to be careful. Number of GA runs, it is given here as a default 10 and population size 150. Okay. So I recommend that you increase it to 50 here and here 300. So computer will run almost 300 poses here and give you the 50 poses here and give you the best pose with best binding affinity. So instead of doing regular 9 pose docking, you perform this accordingly. You increase the population size and GA run. That will give you a far better binding affinity. With regular docking, what you might have find -8 kilocalorie or something. If you increase the GA run and population size, it may increase it to -12 also. So computer will use more resources, your computational time will increase. Maybe you might have seen for regular dockings, it will take hardly 2-3 minutes. But if you increase the GRN and population size based on your computer specification, for one docking, it might take maybe half an hour also. And if your specifications are good, it may complete in 10-15 minutes also. But you will get far better results than your normal docking. Okay. So here, because time is less, so we will shorten this. I will just keep it 5 to show you that how results come. And population size also I'll keep 50, maybe 30 less, and we'll accept it. So that has been done. Now again, we'll go to docking and we'll select the output option. And here you choose the Lamarckian algorithm. It will create the output file. First one was your grid parameter file was .gpf file. Here you have .dpf file. So same name you can give, 1.dpf or anything you prefer, and save it. Okay. So we have saved, and final thing what we need is to run the Autodock. So come to run and run Autodock, and here again browse and select this Autodock 4.exe file. Okay. And in the parameter file, you select the 1DPF and launch it. So it will take little bit time, maybe 2-3 minutes. If there is any question in between, you can ask. So once it performs the docking, you will get one DLG file, .dlg file, and I'll show you how to analyze that file. Whatever I am showing, just it is a basic principle. There are many other things which needs to be incorporated while performing this kind of things. But you at least you follow these things, you will get better results than your regular dockings. Then there is a thing called protocol validation, that whether your software, whatever docking results it has given, whether it is accurate or not, how to check it. What you can do is that, like this structure, what we have taken from the RCSB. So 6LU7, I think. So here you can see how to validate the protocol. It is written that there is a structure and there is an inhibitor, which is already present in this structure. This inhibitor is called as co-crystallized ligand. Okay. So to validate the software, whether the software is giving proper results or not, what you can do is that separate this co-crystallized ligand from this receptor and use it as an individual ligand, and again dock back to this receptor. If the software is able to dock it in the same place where the ligand was in original, and the RMSD, that is root mean square deviation from the original structure, is less than 2 angstrom, that means you can say that your software is giving accurate results and protocol is validated. If the results are deviating, that means you cannot use those results. And that is the main thing which I have seen while reviewing papers. I have seen that almost 90% of the people don't do, and the reviewers ask for the same thing. So this is an important point, that after docking, you need to do the protocol validation also. Okay. So it is still running. So don't do anything, don't click dismiss or anything, it will automatically go. Once the run completes, it will automatically go. There is another easier way by which you can do docking without doing so many things with the help of Pyrex. If time permits, I'll show you that also. Within few clicks, you can complete the docking. Okay, let me tell you one more thing. If suppose, okay, it has already gone, the doc, no, it is still running. Okay, let me show you one more thing. Suppose in this structure which we have downloaded, there is a gap in the structure. There is a missing amino acid residue. Then how do you complete the structure before docking? You need to refine the structure, right? So one easy method, there are multiple methods like modeler and all. For that, you need again this kind of coding system and all those things. It will take time. But easily what you can do is that go to this Swiss model software, sorry, server, Swiss model, and select start modeling, and go to your RCSB file. Okay, RCSB 6LU7. If suppose this structure is not complete, what you need to do, instead of downloading the PDB format, what you can download is FASTA sequence here, the first one. Download, it will open in text format, open in the notepad, and select complete this thing. Because we know that we have only the requirement of chain A, so we will select only chain A from here. Rest of the things we don't require. So what we can do is that we can directly go here in the Swiss model software and paste your target sequence. So I have pasted here and just search for templates. What it will do is that how it will try to fill the gap, the protein structures which are available in the database. There are multiple structures which are similar to each other. Okay, so it will compute that with this structure, which protein, other protein which is complete is similar structure. So it will try to extract those amino acids and fill the gaps here in this structure. Okay, so easily you can complete your structure and you can take it forward for docking. So here I think our docking is complete. So that folder has been gone. So it should generate one DLG file. Let us see whether it has generated or not. Okay, we got here one.dlg file. So we will open in text format. Sorry. Okay, like this you can open in Notepad and press Ctrl S and type RMSD. Okay, when you type RMSD, just search it and you will find here first, not the first one. You go to the second thing, RMSD table, this one, RMSD table. So because I had done a very short docking for a very short time, so it is giving so much less docking binding affinity. Even it is coming in positive also. Okay, that usually doesn't happen. But I just showed you the demonstration, so that's why it is giving such kind of results. If you consider it for very long time with complete docking, obviously it will come -7 or -8. So why negative charges are stable? Because for any complex to be stable, what you need, the energy needs to be reduced, energy needs to be released. So if energy is released, it is a negative term, right? So that's why it is given negative energy. Okay, so the first thing is usually the first rank is usually what you take is the best, obviously is the best binding. If you consider all these runs, you will find that -2.25 is the lowest score. So we know that rank 1 and run 1, we need to see this run 1 is having the minimum binding energy. So we will take this complex only to the MD simulation validation purpose. Okay. So how to extract this complex? We understood that our rank 1 was the best. So how do we extract it? Just close this. The work is over. We can go to file and exit. Do you wish to quit? Okay. And again, open the Autodoc. Now you come to analyze section. Everything is done. Docking is done. Now you come to analyze section. Go to the docking and open this one.dlg file, which which has been prepared after docking, it will show you this warning. Just press OK. So once you have opened this 1.dlg file, what you need to do is that open the macromolecule, it has opened the macromolecule. Then what you can see, what you can check the runs. See, I don't know whether it will be visible or not to you, but as I play this forward, the position of the ligands are changing. If you can see like this. So first it is here. The computer is trying to dock it in different places over the receptor. First, it is present in this section. In the next run 2, it is present in here. Then it has shifted to here. Then here. And then again back here. So I had just given 5 poses. So it could not find better places. So at least you give 50. So it will search for better places where binding affinity is less. So keep the runs around 50. So it will give you good results. So suppose my first run was giving me the best result, as I saw in this table, that my first run was giving the best result, that is run 1, and binding energy is also less, minus 2.25. So what you can do is that from here we need to extract this, select this first run from here, select this open panel to change play option, and show info. So it is showing the first one is binding energy 2.25, right? So we are in the correct run. And just write complex. Here there is an option called as write complex. You write it and give a particular name, maybe receptor ligand final complex. And it will be also in the form of PDB-QT format. So we will save it. That's it. The Autodocs work is over here. We can close it here. Okay, now we have the complex. Now we need to see the visual interpretation, right? So for that we had PyMol and Discovery Studio. PyMol and Discovery Studio are basically your visual tools to identify the bonds and structures. So did we generate? Yes, we have here. See, there is one problem that it is not showing any kind of file format. If this kind of thing happen, what you can do, just rename and add a PDB QT format here. So it will now open up in PyNol. Just double click. See, now the receptor is bound with this ligand in this position. If you want to see properly, you can change the color also. Just click the sequence here. This UNL1, which is separated from this entire protein sequence, that is your ligand. So you can select it here, and from the right side panel, select color, change any color you want, you can give. I have given red here. So you can see that this is your complex. Okay. So what you can do, this is 3D visualization, right? And in PyMol also, we can do better visualization, but that will take another 1 or 1.5 hours to show you how to properly visualize it. So we'll stop it here only. But this is a 3D structure, but we need to see actually that which amino acids are binding to which part of the receptor. So for that, we need a 2D structure of this structure, right? So what we can do is that save it, export the structure, because Discovery Studio cannot open PDB-QT file. So we need to convert it into PDB file. So export structure, export molecule, and convert it to PDB. See, here always default save as type will be PDBX, right? So we need to select PDB here, and it will show your folder Autodoc where you are doing everything, and give it a name, receptor ligand complex. One more thing I would like to tell you, when you do this entire docking process, try to not give any gaps in the name or any kind of symbols or like this underscores. Avoid this, because I have seen multiple times the Python is having problems with this kind of names. So it will give you errors while doing the talking. Keep it simple without any gaps. Okay. So we'll save it here. We have saved, we can close the PyMol. If we want to see the 2D structure, what we can do is that this is our PDB file. We'll just open it in the Discovery Studio. Sorry, it opened directly in PyMol. We'll close it. Open with Discovery Studio. So I have one more thing I would like to tell you, that always when you are publishing your papers or giving your results, use always the white background. Black backgrounds are not suitable for publication. So if you, any kind of, not only this CAD related, any kind of diagrams you give, always give white background in the image or figures, whatever you are giving. Okay. So what you here by default, it will come black. So how you can make it white? Go to scripts and visualization, then publication quality, and it will change it to white. See here also you can see this, right? This is the ligand which is bound. So here in the left panel, you see define the receptor and ligand. The receptor is already defined. Define receptor, receptor ligand complex. It is already defined. So we need to, but define ligand, it is undefined. So to see the interaction in 2D format, we need to define the ligand also. So how we can define it? So if you open this panel, there is something called as ligand groups. You select it, it will select here. You can see it has selected the ligand in yellow color, and just click here, define ligand. It has now selected the ligand. Now there is only one step remaining, that is show 2D diagram in the left panel bottom. Show 2D diagram. So you can now properly visualize that this is your light color things, this is your ligand, and these bright circles are your amino acids from the protein with which this ligand is interacting, and with which kind of bonds it is given in the interaction legend. Okay, light yellow is light green is van der Waals forces, and as much as your conventional hydrogen bonds will be more, that much your complex is stable. Okay, van der Waals forces are the weakest forces. Then you have certain kind of hydrophobic interactions also, pi alkyl, pi alkyl. So all these things you can understand. You can later read that which kind of bond is for what purpose and all those things. So that's how you can visualize. And if you want to save it for publication purpose, just click it here and file and save as. Here you will find option save as type, image type, anything, PNG, JPG, whatever, you can just select and it will save. And suppose you want to save the 3D structure also from the Discovery Studio, what you can do is that here choose ligand interactions. So this kind of structure will open up in the 3D section. And if you want to see that what kind of hydrogen bonds are there, means hydrogen acceptors are there or hydrogen donors are there in which part of the receptor, it will give you a colored map here. So from this also you can understand. If you suppose want to label it, just right click, label and add, and from the object you select amino acid. From the font you select maybe Arial black, and color you can choose black and apply. So now it is showing in 3D format also that the ligand is connecting to which amino acids of the protein. Suppose you want to see that which part of the protein from the 3D structure is corresponding to which part of the 2D structure. Just with your mouse, you make a circle, yellow circle, and it will show that which part you have selected here. Suppose this part I am selecting. So see here, this region, it has highlighted in yellow. That means this part is corresponding to this. You can see the difference of the aromatic rings here. Okay. So these are certain things, not only these things, you can go through this software and you can find multiple visualization techniques, and you can make it better as per your convenience. Okay. So that's what I wanted to show you today. and maybe time has crossed to show you the Pyrex. Maybe some other day I can show you. So that's it from my side. Thank you. And if you have any questions, you can come up with. Is validation required if we are using online docking sites like CBDoc, SwissDoc? Yes, for everything validation is required. Even what we show today is your protein ligand docking. You can do protein docking also. For that, you can use online softwares like ClassPro and HDDoc or something like that. You can just find over Google, you will find it. Anything else?
Sir, you have a question. If we are using online docking sites like CityDoc, SwissDoc, is the method validation required?
Yes, I think already I have given the answer. The validation is required. You can use any software, but validation is definitely required. So one thing I would like to show you, if you want to see any kind of journals where you can publish. So these are some of the journals where you can publish your total computational works. These are some of my papers published over last few years and of different publishers. You can go through it. And that's it from my side. Is there any site from where we can prepare our own ligand if the ligand data is not available? Site is not required. If the ligand data is not available, you have that information or knowledge, what kind of structure you want to prepare. You can just use ChemDraw and you can save it in formats which you can utilize in CAD softwares. ChemDraw is available in the Google, and I think it is there in the BPharm syllabus also in the last semester. So ChemDraw is available to everyone. You can draw structures there and you can use that as per your wish. But the problem is that you can draw any structure, but the problem comes that whether that structure is synthesizable or not. That is the point. Maybe any structure is giving you good binding affinity, but whether the structure is synthesizable or not with the current technology, that is one question. So you need to consider that point while drawing a structure.
Thank you, sir. Any participants having any queries, you can drop your questions in the chat box or you can directly ask to the speaker, sir.
Is the energy minimization of the structure required? Yes, energy minimization of the structure, if you perform, that will be better. As I said, you can use both Chimera also, and there is one more software called a Swiss PDB. You can find tutorials over YouTube also for Swiss PDB. It is very simple. Just drop the structure. It will automatically minimize when you click on the minimize structure. That is for protein structure, and for ligand structure, you can use ChemDraw to minimize. You can use Pyrex also to minimize the structure. Minimization is required because what happens when you get the raw structure, the energy inside the structure is high. So flexibility will also increase while docking. Okay. So if you minimize the structures are stable, it will give you good binding affinity. For MD, which software is used? For MD, if you have like license from your institution, if you can buy it, then Schrodinger is best. And if you cannot, but you have a better system, then you can use Gromax. Gromax is a free software, but it will require coding knowledge little bit.
Thank you, sir. Any participants having any questions? Sir, is GATE software required for simulation?
Is paid software required for simulation? Like it is up to you. You can use the paid one also. That is Schrodinger suite is paid and Gromax is free. So, but Schrodinger is easy to use. For Gromax, you need some little bit coding understand because it runs in Linux. Both the Gromax software and the Desmond will run in Linux. It will not run in Windows. So you need a Linux system and little bit understanding of the Linux. That will be enough.
Sir, we have another question. Which are the free version of for simulation?
For simulation, you can use any version of the Gromax, but the problem is that you cannot use your regular computers. It will take a lot of time for 1 nanosecond. So preferably journals ask you for at least 100 nanoseconds. For that, you need a proper system. It will cost you around, with good graphics card, it will cost you around 2 lakh rupees. So if you can afford that system, then you can use Gromax for free. The software will not cost you, but the setup will cost you. And if you are using Schrodinger, it will be easy to use, but it is highly costly. So only institutions with good funding can avail it.
Sorry to interrupt, sir. I think you got paused.
I think, am I not audible now?
Yes, sir, you are audible. Thank you, sir. It was such
Okay.
an informative session on computer-aided drug design. So now we'll move towards next. So I would like to request Professor Dr. Subhasis Deval, sir, Principal of Bharat Pharmaceutical Technology, to present the certificate to our speaker, Dr. Kunal Bhattacharjee, sir, virtually.
Sir is there. I think
No,
some.
sorry.
I think, sir, got
Sorry.
some network problem.
Forty seconds, 30 seconds.
Sorry for the inconvenience. We have some technical issue.
Sorry, due to heavy rain, we have power interruptions.
So
I
okay.
would request Professor Dr. Subhasis Deval, sir, to present the certificate to Mr. Dr. Kunal Bhattacharjee, sir, virtually.
First of all, on behalf of Bharat Pharmaceutical Technology, also the organizing team, I thank Dr. Kunal Bhattacharjee for such a wonderful presentation, and also the way sir has demonstrated the thing. That is really very wonderful. Many participants in the chat box also, they are writing nice presentation, effective presentation. Please share the video link, because it is not possible that just within a 1 hour we can see or understand and summarize all the things. But then also, the way Dr. Bhattacharjee has presented the thing, I salute for this. And also for the participant, we have our YouTube channel, and in the YouTube channel you will find out all the videos. Like it will, like if you want to refer the things. He is the youngest resource person we have. And when we decided, no, when we are deciding that we will go for such type of FDP on AI and all, and I thought that I must invite Dr. Bhattacharya for this session, because I have seen him personally when I was in Royal Global University. He was doing PhD over there, and the way he present, the same way, like you find out, this is very difficult, but Sir has presented the thing very easily and very smoothly. So all of the teachers, we should have such type of talent, that how to present the thing very skillfully, and that also we should learn. So thank you very much for your time, and also giving most of the knowledge to all the participants and also to our faculties. I request
Thank you,
Pralay
sir.
sir to share the certificate. So on behalf of the Bharat Pharmaceutical Technology, I request you to accept the certificate virtually, and
Thank you,
hope
sir.
we'll meet. Normally we are meeting, but maybe for professional purpose or some conference, we'll meet again physically also.
Yes, sir.
So thank you very
Sure,
much
sir.
for everything.
Thank you, sir.
Thank you.
Thank you, sir. Now I request Ms. Arunima Das, Assistant Professor of Bharat Pharmaceutical Technology, to deliver vote of thanks. Good afternoon, respected Principal Professor Dr. Shubhashish Devnath, respected Speaker Dr. Kunal Bhattacharya sir, esteemed participants, and my dear colleagues. I, Arunima Das, Assistant Professor of Bharat Pharmaceutical Technology, have the honor and privilege for delivering the vote of thanks on behalf of organizing team of the EFDP on immersive technologies and artificial intelligence in pharmacy education, building the future of learning. First and foremost, I extend my heartfelt gratitude to our distinguished speaker, Dr. Kunal Bhattacharya sir, for his insightful and enriching lecture on computer-aided drug design, principles, tools, and applications in modern drug discovery. Sir, your presentation was not only highly informative, but also inspiring, showing us the immense potential for computational approaches in shaping the future of drug discovery. Your valuable thoughts and comprehensive presentation have truly enriched our understanding of CAD and different CAD tools. We are indeed fortunate to have benefited from your expertise and insights today. Thank you so much, sir, for this wonderful session. I would also like to thank all the esteemed participants, the faculty members who have joined us from different institutions across the country. Your active involvement and enthusiasm have greatly contributed to the success of this event. I would also like to acknowledge the tireless efforts of the organizing committee, the technical team, and everyone working behind the scenes to ensure the smooth conduct of this event. Lastly, I thank each one of you present here today for making this session meaningful and impactful. We look forward to your continued participation in the upcoming sessions of this FDP. Once again, thank you all and have a wonderful day. Thank you, ma'am. Now I
Okay,
cordially thanks all the participants for your valuable presence. We will share the evaluation and feedback link in the WhatsApp group, and you are requested to submit it by today. Thank you. Have a wonderful day.
Thank you, everybody.
Thank you, sir.
Recording stop.